Doo-doo-doo-doo! This is the transport video by Lexi Hemming, Clara Newton, and Ryann Melville. The lipid bilayer, otherwise known as the membrane, is made up of a hydrophilic phosphate and glycerol head and a hydrophobic tail with two fatty acid tails. Some are saturated with single bonded carbons, causing them to be straight, and some are unsaturated with one or more double bonds between the carbons, causing them to have kinks or bends. The hydrophilic heads are polar, and the hydrophobic tails are nonpolar. Diffusion is the movement of a substance on its concentration gradient. A concentration gradient is whenever the concentration of a dissolved substance is higher on one side of the membrane than the other. The substance moves from a higher concentration to a lower concentration to create equilibrium. This is passive transport because the cell does not expend energy when the substance is diffused across the membrane. Facilitated diffusion is a type of passive transport through proteins. The polar molecules and ions use this because they can't get through the lipid bilayer. This happens because they are hydrophobic. There are two types of facilitated diffusions. One uses channel proteins and the other uses carrier proteins. Channel proteins have ion channels or gated channels. They open or close in response to electrical or chemical stimuli. The carrier protein has a solute that binds to it and then changes shape to translocate the binding site in the attached solute across the membrane. Osmosis is a diffusion of free water across the membrane. Water diffuses down the concentration gradient from a high water concentration to a low water concentration to make equilibrium. Tonicity is the tendency of a surrounding solution to cause the cell to gain or lose water. This is affected by the concentrations of solutes. An isotonic solution is when the solutes have equilibrium. An animal cell will neither gain or lose water in an isotonic solution. A hypertonic solution is when there is a higher concentration of water on the outside of the cell than the inside. An animal cell in a hypertonic solution will lose water and shrivel and might die. A hypotonic solution has a higher water concentration in the cell than the outside. An animal cell in a hypotonic solution will gain water molecules, causing it to possibly lyse or burst. A plant cell in a hypotonic solution has balanced water concentration. Water moving into a plant cell causes it to swell against its cell wall. This is turgor pressure. In an isotonic solution, plant cells become flaccid, which means limp and droopy. Plant cells in a hypertonic solution go through plasmolysis. This is when the plasma membrane pulls away from the cell wall as the water comes, and then the cell shrivels. Active transport uses energy to move solutes across their gradients. The cell uses energy because it has to transport solutes across the membrane. It goes from a lower water concentration to a higher water concentration. This is done by a protein pump against a concentration gradient. Examples of this are the proton pump and the sodium-potassium pump. The proton pump actively transports proteins out of the cell. ATP is a terminal phosphate group which supplies energy for most active transports. The sodium-potassium pump exchanges sodium and potassium across the animal cell membranes, creating a higher concentration of potassium ions and a lower concentration of sodium ions within the cell. Bulk transport requires energy to transport large biological molecules packaged in vesicles across the membrane. A vesicle is a small structure within a cell or extracellular consisting of fluid enclosed by a lipid bilayer. Endocytosis carries out bulk transport. This is when a section of the plasma membrane sinks inward and pinches off to form a vesicle containing material that used to be on the outside of the cell. Phagocytosis is a form of endocytosis where it engulfs a food particle or other large particle, creating a vacuole that then fuses with the lysosome. Penocytosis is when droplets of fluid outside the cell are taken into the cell in small vesicles. Receptor-mediated endocytosis enables the cell to take in specific substances from outside fluids. Ligand is a term for any molecule that binds specifically to a receptor site on another molecule. Exocytosis is the process in which the cell secretes certain large molecules by the fusion of vesicles by the plasma membrane.